I've been discussed for the day what is mutation and gene regulation.
What is mutation? A sudden change in physical or chemical structure in genes or genetic material, or more specifically, sudden changes in the nucleotide sequence of DNA. May occur in somatic cells, are not passed to offspring. May occur in gametes, eggs and sperm, and be passed to offspring. The result due to mutation is immediate. What is mutation? A mutation is a change in a DNA sequence. Mutation can result from DNA copying mistakes made during cell division, exposure to ionizing radiation, exposure to chemicals called mutagens, or infection by viruses. Causes of mutation. Mutation arise spontaneously at low frequency owing to the chemical instability of purine and pyrimidine bases and to errors during DNA replication. Natural exposure of an organism to a certain environmental factors, such as ultraviolet light, chemical carcinogens, aflatoxin B1 also can cause mutations. We have the three types of mutations. The first one, the base substitutions. Single base substitutions are called point mutations. Recall the point mutation global which causes sickle cell disease. Point mutations are the most common type of mutation, and there are two types, the transition and transversion. Transition. This occurs when a purine is substituted with another purine or when a pyrimidine is substituted with another pyrimidine. Transversion. When a purine is substituted for a pyrimidine or a pyrimidine replaces to a purine. Deletions. A deletion resulting in a frame shift results when one or more base pairs are lost from the DNA. If one or two bases are deleted, the translational frame is altered, resulting in a garbled message and non-functional product. A deletion or more bases leave the reading frame intact. A deletion of one or more codons results in a protein missing one or more amino acids. This may be deleterious or not. And last, the insertions. The insertion of additional base pairs may lead to frame shifts depending on whether or not multiples of 3 bases pairs are inserted. Combinations of insertions and deletions leading a variety of outcomes and also possible. Example of mutations. Sickle cell anemia is caused by a substitution in the beta-hemoglobin gene, which alters a single amino acid in the protein produced. Change codon to one that encodes the same amino acid and causes no change in the protein produced. These are called mutations. Chemical factor of mutation. Cigarette smoke contains dozens of mutagenic chemicals, nitrate and nitrate preservatives in hot dogs and other processed meats. Barbecuing creates mutagenic chemicals in foods. Benzoyl peroxide, common ingredient in acne products. We have the gene regulations. Regulation of gene expression or gene regulation includes a wide range of mechanisms that are used by cells to increase or decrease the production of specific gene products. The process of gene regulation. Gene regulation is the process of turning genes on and off. During early development, cells begin to take on specific functions. Gene regulation ensures that the appropriate genes are expressed at the proper times. Gene regulation can also help an organism respond to its environment. Example of gene regulations. One of the jobs of the liver is to remove toxic substances like alcohol from the bloodstream. To do this, liver cells express genes encoding subunits or pieces of an enzyme called alcohol dehydrogenase. This enzyme breaks alcohol down into a non-toxic molecules. In gene regulation, we have the induction and repression. In induction, we have the active repressor, operator, block, inducer, and free. In repression, we have inactive repressor, free, compressor, and block. In gene regulation, we have the 1 and 2, initiation of transcription, 3, mRNA splicing, 4, mRNA protection, and mRNA processing, 5, initiation of translation, 6 and 7, protein processing degradation.